Hi girls, it's Mrs. Vasquez. Here is our video for homework. If you remember in class, we last talked about incomplete dominance, right? So this exception to Mendel's rule. We talked about Andalusian chickens. We also talked about snapdragons, where basically the difference between incomplete dominance versus everything we kind of learned with Mendel and his P plans is that in incomplete dominance, neither allele is actually dominant over the other. Right? So in snapdragons, if you were heterozygous, meaning you inherited a capital and a lowercase from two parents, neither red nor white is going to show that dominant trait, and instead they're both shown incompletely. So you show a blend of the two, and that's where we see pink as our color. Now, your homework was to do some practice problems showing incomplete dominance, and that was number 6 through 10 in the packet. Remember, the packet is attached or uploaded into your Veracross homework, so make sure to check that and to print it out or copy the questions onto loose leaf. But I thought we'd start first. I copied one of the questions, I think it was number 6 on your homework, the Snapdragon question. I changed the question around a little bit just to give you another opportunity for extra practice, but you'll notice some similarities to number 6 on your sheet. So let's read it together. It says, in snapdragons, flower color is controlled by incomplete dominance. The two alleles are red and white. The heterozygous genotype is expressed as pink. So heterozygous means that the pink flower genotype has to be big R and little r. A pink flowered plant is crossed with a red flowered plant. What is the probability of producing a pink flowered plant? So what I want you guys to do is I want you to pause this video, get some scratch paper, figure out what are the genotypes of the two parents, write them and set up the Punnett square, fill out the Punnett square, and I want you to answer that last question. What is the probability of producing a pink flowered plant? Okay, so pause the video. Okay, so hopefully you've unpaused and we're going to go over the answer now. So parent number 1 is pink, so we know that parent number 1's genotype has to be big R, little r. So I'm going to put that parent up here. Again, remember, it does not matter which side you put the parent. Second parent is a red flowered plant, so we know that the red allele is big R. So a red flowered plant has to be big R, right? It can't be big R, little r, because then it would just be pink. It has to have both copies of the red allele in order to show that red trait. So now we're going to distribute. We'll do the red parent first. And now I'll distribute down with the pink parent. Okay, now we have our Punnett square. So now we're answering the question, what is the chances of having a pink flowered plant? So how many boxes are showing a big R, little r? So not this one, not this one, this one, and this one. So the answer is going to be 2 out of 4, or since it's asking for percentage, 50%. And that is going to be the correct answer for this question. Okay, so check over your work. If you have any questions, make sure you kind of re-listen to that video to make sure you see like how I got each of those genotypes in Punnett square. And we can always answer those questions in class too. Okay, so now we're going to be taking some notes. We're really not taking too many notes, it's just this one slide, but I wanted to introduce you guys to this other exception to Mendel's rules. So I'll come back to this in just a minute. Remember in our last class, we talked about patterns of inheritance, right? Other patterns. So what did we observe that are kind of exceptions to what Mendel discovered? And so in class, we talked about incomplete dominance, and today we're going to talk about codominance. All right, so we're going to see that there's a lot of similarities and differences between codominance and incomplete dominance. Okay, so one similarity is that just like incomplete dominance, neither allele is going to be fully dominant or really fully recessive. You're going to see both alleles kind of working together in an interesting way when you are heterozygous for that trait. Okay, another similarity is that like incomplete dominance, you're not going to see like a third or new phenotype unless you're heterozygous. So we're going to use chickens again. So these are not our Andalusian chickens. This is another kind of chicken. But chicken feathers and other bird feathers do sometimes show codominance too. So what happens, let's see, we have big B is going to represent black feathers. And we'll stick with B prime like we did with the other chicken example. B prime is going to represent white feathers. Okay, so if you are a black feathered chicken, that just means your genotype is big B, right? You have only the black allele. If you are a white feathered chicken of this species, your genotype is going to be big B prime, big B prime. Okay, no question other than those two. So, but the problem or the new phenotype, it comes when you're heterozygous. And this is very similar to incomplete dominance, where if you inherited a plain B from a black feathered chicken and you inherited a plain B dash from a white feathered chicken, you are not going to show black or white alone. You are going to actually show them together. You're not going to show a blend of them. So in Andalusian chickens, remember black and white were blended together and you saw gray. In codominance, you're going to see them dominating together and you're going to see black and white actually showing up at the same time. And so in here, it's a little bit hard to see, but if you look really closely, let me see if I could zoom in, you'll see that they have what we call a checkered pattern where you actually see black and white. So sometimes you see this in stripes, you see it as blotches, you see it as spots. And so the word codominance, if you think of the word co. So co means together, the prefix. If you think of, let's say, if you've ever been on a team and there was a co-captain, that means you probably had 2, maybe 3 captains that were acting as captain together. They were leading the team together. They were both captains. They were both dominating or leading the team. In codominance, both of the alleles, when you inherit both of them, so if you're heterozygous, both alleles are going to be dominating together. They're both going to be expressed. And that's why you sometimes see multiple colors show up if you're looking at color as a trait. Okay, so one similarity between codominance and incomplete dominance is that it is always the heterozygote that is going to show that new or interesting phenotype. Okay, and in this case, in codominance, it's not going to show a blend. It's going to show both traits at the same time. Now, color is usually the easiest example to visualize, like in this example over here, right? So this is another example of a flower. These are not snapdragons, different type of flower. And we have a pink allele and we have a white allele. But if you're heterozygous and you have a pink and a white, you are actually going to show both pink and white at the same time, not a blend of the two. Okay, so this is an example of codominance. Another example, though, of a trait that's not really color is actually our blood type. So some of you guys may know your blood type. Maybe your parent remembers what blood type you are and has told you. If you don't know your blood type, you can easily find out the next time you have your blood drawn. You can ask your doctor, or if you've ever, I don't know if you guys are too young to donate blood. I don't remember, but if you've ever donated blood or you plan to, they'll also tell you your blood type there. And there's 4 possibilities of blood types you can have. And everyone has one of these 4 blood types. And you can either be blood type A, blood type O, blood type B, or you could be blood type AB. And this is a case of codominance. We're going to learn in class Monday and Tuesday how blood type actually works and what those A's and those B's and those O's really mean. But if you're blood type AB, that means you're showing the A and the B type blood at the same time. And that actually gives you a new phenotype. And so this is an example that's a little bit hard to visualize because it's not a color example, but also an example of blood type. And we're going to learn that blood is actually very interesting too, just in its own way, also because it has more than 2 alleles. So everything we've talked about so far, even if it was incomplete dominance or codominance, everything usually has 2 alleles, right? Big B and big B prime. You may have a third phenotype, but the alleles always stay as 2. Blood type's different though, because blood type we're going to find out has 3 alleles, which makes it very interesting. So one thing that I hope, and if you haven't started thinking about it as you're listening to this video, I want you to start thinking is how is codominance different than incomplete dominance? We're going to see some big differences in terms of how that third phenotype is actually expressed, but you probably have been noticing some similarities, right? The fact that the heterozygote is the one that's showing those third phenotypes. The fact that neither allele is being dominant. And of course, the fact that I've given you chicken examples for both. But codominance and incomplete dominance happen in different types of plants, different types of animals. We see it in humans. And one way, oops, sorry, I forgot to delete that. I'll do it with you guys. One way to kind of visualize the difference between them is I put over here in incomplete dominance. If you had a red flowered parent and a white flowered parent, your heterozygous offspring are going to show a blend. Think neither allele is completely showing, it's incomplete, so it's like a blend of the two. Whereas in codominance, you could still have same parents, a red allele, a red parent, and a white flowered parent. But in codominance, you're going to see both of those traits being shown at the same time. That's why sometimes you tend to see spots, you tend to see blotches of color. Okay, so you can have one flower that shows incomplete dominance for flower color, and you can have a different species of flower that shows codominance. So it really depends on the organism that you're studying. We tend to use plants a lot just because color is a really easy way to visualize the differences, but we see it with a lot of other traits too, even just in terms of length of certain body structures, which we'll talk about more examples on Monday and Tuesday. All right, so when we come to class, we'll review these two. We'll do some more practice problems. The last part of your homework for Monday and Tuesdays, I would like you to go back to that packet that you did the incomplete dominance practice questions 6 through 10. And now I want you to do numbers 1 through 3. Go back to that first page. Number 1 is pretty simple. It's not a word problem. It's just having you compare incomplete dominance and codominance. But numbers 2 and 3 are going to give you some practice to try and do a codominance Punnett square and word problem. All right, guys, have a wonderful weekend. Email me if you have any questions, and we'll go over all of this on Monday and Tuesday.